Hi, today I'm going to talk to you about how Zegami can be used in high throughput screening. This was a paper published in Nature Communications in 2015 where we were studying how a molecule called ATRX was being suppressed. ATRX is an important molecule in a type of cancer that is associated with the lengthening of telomeres. So in this collection we've generated a series of montages to represent the way we represent this data. And we're interested how different protein molecules in the nucleus come together, and these protein molecules cause ATRX to be suppressed. So in Zegami you can choose any graphical representation to represent your screen. In this case we've chosen this representation, which we split into 8 panels, where you can see the original cell nucleus with the different molecules in here, and we've got TRF2 as one molecule and PML as another molecule, and when they come together you can see they make this yellow colour, and that's called co-localization. And we can look at the different ways these come together. There's the types of co-localization here. These are represented, you can see these two overlap, these two red spots overlap here, causing them to co-localize, and you can see that here as well. And you can also see the grayscale representation of the image. And having access to this level of detail is extremely valuable when you're verifying the results of a screen, and especially when you're using experimental computational pipelines to extract large amounts of data from a biological system. Because biological systems are inherently variable and the way you capture the information is also variable, they often have different artifacts and outliers. And often when you're developing these methods you don't know what to look for. For example, if we zoom out here, we can see there are actually 2 weird sort of artifacts that have come out of the screen, and that's down to the fact I've used a type of enhancement, which has meant the number of red foci has been massively kind of overcalled, and they're kind of all merged together, whereas there isn't really anything to kind of see here. So one thing we can do in Zegami, we can enter a mode where we can tag these 2 bits of data out, and we can say these are bad ones. So that's now tagged out, and then we can exclude these from a future analysis by just going into the filter panel and looking at the tag section and say we only want to look at the ones that aren't labelled as bad. So you can see these have gone now. So the other nice thing, we've got a range of chart types we can look at. So if we want to look at potentially how the number of foci or dots have been called, we can look at this in a sort of like what we call the graph view. So this shows a bin distribution of the number of points, and we're looking at the red channel here. So for example, we can kind of zoom in over here. And we can see there's again almost like an outlier where there's been many spots have been called, whereas actually there's probably only a few foci in there, and these could also be excluded from the screen in a similar way. So the number of other things you can do, you can look at, for example, how the number of red foci compared to the number of green foci and the distribution there. So we can see here that there's a large amount of red foci compared to green, and we can look at some of these kind of disproportionate outliers here where you can see you've got many red versus the green. Again, it's another nice way of being able to check these particular results. And finally, we can look at, say, things like the distribution of these data under the effect of different drugs. So for example, if I go to this sort of graph view again, I can actually look for the effect when it was in doxycycline, and I can order the number of rows by the number of co-localizing foci, and we have effectively a jitter plot here where we can actually zoom in and look at the extremes really easily rather than it being a static figure. So the other nice thing about this is we can make this a public collection and share this. And this is what we did in the paper. We actually took the 16 or so screens and we made these available as hyperlinks, so the collaborators and reviewers can look at every aspect of the system rather than just cherry picking the best results. And that's important because it means research can be transparent and shared to reduce the reproducibility crisis that's often happening in research.